All right. Unfortunately, I just realized I had not hit the record button. So I'm hoping you guys were able to take notes and you were here in person. And that's why being in class in person is so highly recommended, right? Uh, well, it's recording from this point onwards, but the initial portion would not have recorded unless you recorded it at your end with your cell phone or something. So,
And what was teratogen again?
teratogens are substances or chemicals that cause birth defects in the developing embryo, like nicotine, like alcohol, drugs, drugs of abuse, that is, okay? All right. Let's see, there, anencephaly and spina bifida, we talked about the different types. Okay, so these are the different structures in the embryo that develop into some of the adult structures of the brain, such as the prosencephalon, okay? This is the forebrain, the front part of the brain. The forebrain is called the prosencephalon, as you can see here in the picture. It's blue in color. The midbrain is mesencephalon, and the hindbrain, the back, the hindbrain here is called the rhombencephalon. So all of these are labeled in this picture. And this is a 4-week-old embryo, as we can see. All right, by the 5th week, some more structures become clear here. And such as what is called the telencephalon. So if you see here in the picture, the telencephalon will eventually become the cerebrum, all right, when the child is born. Diencephalon, that is an important region. The diencephalon turns into the thalamus, the hypothalamus, and the epithalamus, which is your pineal gland, basically. So all 3 of those thalamus named structures basically come from the diencephalon. The mesencephalon in the picture will eventually turn into the midbrain, and midbrain again is a part of your brainstem. The metencephalon, okay, again follow along in the picture here, the metencephalon in green color, it becomes the pons and the cerebellum. The myelencephalon becomes the medulla oblongata, which is the inferior most part of the brainstem. So some of the structures and what do they eventually turn into the adult brain. So this is what we are focusing on basically. This is what a well-formed human brain actually looks like in between 13 to 26 weeks of development, as you can see. So it's very much the same as what you would see in an adult. The same structures are seen here. All right, there you go. So this is brain at birth, okay? Again, as we mentioned before, the human child is born with a very large head to body ratio, right? A large head and a small body comparatively. And that has to do with the fact that much of the brain growth and development takes place after birth, okay? So we talked about something called neoteny, N-E-O-T-E-N-Y, neoteny. And neoteny basically means that humans are born in a very immature state, okay? Not only physically, but also mentally. So we literally continue to grow and learn and develop in terms of brain development, you know, from the cradle to the grave. Nowhere is it as apparent as in brain growth. Probably the only species, we are the only species that have such a almost an infinite ability to learn and to grow, okay? And so that explains the extremely large head to body ratio that we are born with, okay? Neoteny is the word again, N-E-O-T-E-N-Y. Okay, gray matter versus white matter. What's the difference? Gray matter is simply your cell bodies, okay? No myelin sheath here. The cell bodies and the unmyelinated neurons, they make up your gray matter. So even when they talk about it in general terms, they say use your gray matter. So what are they actually referring to? Use your thinking skills, which means fire some of your neurons here. So that reminds me, so we might run out of time and then I will reconnect. So please stay on the line. I'll pick things up from where we leave those off. Okay, let's see. I need one more person to let in. Okay, there. All right, so, and then, so what is the white matter? White matter is myelin sheath. So all of your axons that are myelinated, that have that insulation, the myelin sheath around them are myelinated. Okay, you tell me which ones are the fast ones, gray matter or white matter, and why?
I would say white matter because it's the one sending the electronic signals and it's myelinated to make it faster.
Good. And would you mind mentioning that process? What is it called? What type of conduction is that? when the electrical current actually jumps from node to node rather than traveling the entire length of the axle.
Let me look at my notes.
Good. So I'm hoping everyone is doing what Sarah is doing, taking notes. That's great. And also why you should be, even though I will be uploading the lectures on Edmodo, you should be physically present too, because see, I forgot to hit the record button. Okay, maybe I did it willingly, who knows? But if you were here and you were taking notes and recording with your devices, then you're all caught up, okay? Right, so note taking, yes, that is excellent. All right, did we find it?
No, not yet. I have a lot of notes. I'm sorry. I don't remember what it's called.
I hear
Is
you.
it the action potential voltage?
You're right about that. It is action potential, no doubt. But the type of jumping action that you see with the myelin sheath is called saltatory conduction. Saltatory conduction, okay? So it's on the slides, hopefully somewhere in your notes as well. There you go. Here's your brain, okay? Now look at the picture. Tell me, where do you see the gray matter located at? Is it on the outside of the brain or in the deeper parts of the brain? I'm talking about the gray matter.
It's in the outer cortex, but it's also in the inner nucleus, right?
Good, good, good. So two places, yes. It's in the outer cortex, as you can see, this thin strip of gray matter. Those are neuron cell bodies that you can see here. And then as Sarah again rightly said, if you look at some of the nuclei, deeper ones, basal nuclei is what they're called, you see some gray matter there too. The white matter, which is myelin sheaths, again, myelinated neurons, they are deeper to the gray matter here. You can see it on the deeper, in the deeper regions of the brain, okay? Now, the thing to remember is, in the spinal cord, it's the exact opposite. So in the spinal cord, the white matter is on the outside, and the gray matter is on the inside, okay? So it's a reverse situation once we get down to the spinal cord. Could anyone tell me why that happens? What accounts for the fact that the gray matter is on the inside and white matter is on the outside in the spinal cord, as opposed to what you see?
I would say because it's sending the nerve signals out from the spinal cord.
Yeah. So why this reversal of arrangement? Any structural feature
Protection?
Say that again?
Protection?
No, no, well, protection doesn't account for why it's the opposite. So what does account for it is, remember, the brain nerves crisscross. They crisscross, okay? So the right half of the brain goes to the left side of the body, the left half of the brain supplies the right half of the body. It's contralateral, right? So the nerves actually crisscross like this. And that is called decussation. D-E-C-U-S-S-A-T-I-O-N, decussation. That word is on the slides here too, so you'll find it sometime. Decussation of the fibers, okay? Crisscrossing. And we all know that if you have a stroke in the right half of your brain, guess which part of the body gets paralyzed? The opposite side, right? And vice versa. So it's contralateral supply. And that is why the spinal cord has the white matter on the outside. It's the opposite as compared to the brain, okay? All right, so something to remember. There's your spinal cord. You can see it in cross-section. What type of a cross-section are you looking at here? This is a transverse section, okay? So I'm sorry, this is not the spinal cord. We are at the cerebellum, okay? Cerebellum is the mini brain, the little part of the brain that projects from the inferior side. So what are some of the different components of the cerebellum? So this is the cerebellar cortex. So again, gray matter on the outside, very much like the brain, the rest of the brain, okay? And then you have the internal white matter in the cerebellum. And then you have what is called the arbor vitae. You'll see that term too, arbor, A-R-B-O-R. That stands for tree, okay? And vitae means life. So arbor vitae literally means tree of life. And that is what this arrangement of gray and white matter in the cerebellum is referred to as, okay? And again, what does the cerebellum do in general? Muscle memory and muscle coordination, as we mentioned before. So something to remember. Now that's the spinal cord, okay? So if you look at the transverse section of the spinal cord, you see that the gray matter now is on the inside, while the white matter is on the outside. There's the spinal cord, all right? That was the medulla oblongata, which is still considered part of the brain, same arrangement. By the time you get to the spinal cord, it's the opposite now. The gray matter is in the shape of the letter H, English letter H in the middle. And the white matter is on the outside here, okay? So there you can see the gray and the white matter of the spinal cord as well. So what are the four major regions of the brain? So you have
The frontal, parietal, occipital, and temporal.
Well, those are all parts of the cerebrum, right? Those are the cerebral cortex.
Oh, okay.
It's asking about, so cerebrum is one part. What are the other three?
The cerebellum, the brainstem, and the diencephalon or something like that.
Excellent. Yes, that is it. So the cerebrum, the cerebellum, the diencephalon, which includes the epithalamus, hypothalamus, and thalamus, and the brainstem, which includes the medulla oblongata, the pons, and the midbrain. That is right. How does the neural plate form a neural tube? The ectoderm cells kind of dip inwards. We saw that it forms a groove. The tube comes together, and that's how you form a neural tube, which will form your spinal cord. Identify the 5 secondary vesicles. So telencephalon, mesencephalon, metencephalon, and myelencephalon, the ones that we saw before. Where is gray matter located within the cerebrum? It is on the outside. And how about the spinal cord? It's on the inside. It's the exact opposite. All right. Okay, next up, we are talking about the cranial meninges, right? So what are cranial meninges? Meninges are these connective tissue layers or membranes that protect and cushion and cover and nourish the brain and the spinal cord, okay? Those are the 3 meninges. And that is why when you get an infection or inflammation of any of those layers, it's called meningitis. All right, inflammation of the meninges. So meningitis is the actual problem, an actual infection and inflammation of one of these 3 membranes, okay? All right, so what are those 3 layers? The outermost is called the dura mater, okay? Mater, M-A-T-E-R, not matter, mater. That's an important difference. In Latin, mater means mother. So these are the mother membranes of the brain, okay? So the outermost and the most resilient one is the dura mater, underneath which is the subdural space, okay? A very potential space underneath. Then you have the second layer, which is called the arachnoid mater. Then below the arachnoid mater is the subarachnoid space. And then the last and the most fragile and delicate one is the pia mater. Okay, so 3 meninges, dura mater, arachnoid mater, and then the pia mater. And you can see all 3 here in this picture, okay? Now, one important question. One of these spaces, right, we talked about the subdural space and we talked about the subarachnoid space, contains the all-important cerebrospinal fluid, or CSF. Which space would that be?
The subarachnoid.
Subarachnoid space is the right answer. That is where you have the cerebrospinal fluid present at, okay? And you might be wondering, okay, so why does it matter? Well, it matters because if someone comes to you with what seems like a case of meningitis or encephalitis, right? A college student, a TCC student for that matter, who's been suffering high fevers and is reacting to bright lights, has photophobia. Photophobia means reactivity to bright light, has